Yes, we'll start our session. First of all, very good evening to everyone. And in Indian time, now it's almost evening. In Kenya time, it's afternoon. So I like to say good afternoon and good evening to everyone. And today is Aqua Doctor Solution Second Foundation Day. And I just finished my Foundation Day celebrations. And as we every day we organize on, especially on Sunday, this particular time of evening 5 to 6, 1 hour session exclusive on aquaculture and fisheries. And this program is supported by National Fisheries Development Board and managed Hyderabad Government of India. Our aim and objective is through this kind of program, we are starting 30 Sunday, 30 speaker, 30 session on aquaculture and fisheries. And today we have today we are the 6th session of our these 30 sessions, and luckily we are also very happy to have with us Dr. Eric Ogle from Kenya, who is also very renowned in his field of biofloc and larval food productions and the larviculture, and we are very good friend. And on my request, on the Foundation Day of Aqua Doctor Solution, we are also getting expert from outside of the country to especially to address on these sessions. And today, Dr. Eric will be addressing us how we can take biofloc technology on the aspects of larviculture, because we understand that larviculture is a very important part. Most of the hatchery, most of the live food production unit in terms of fish or in terms of shrimp culture is considered. So he will be addressing us much more deep knowledge on that, and I like to hand over the sessions and the platform to Dr. Eric to take it further way, so that we everyone can enjoy the sessions and we can learn from his experience and expertise field of this sector. Thank you, Eric, please.
Thank you so much, Dr. No Baman. Thank you so much indeed. I'm very happy to be part of this success in your company, and I vividly remember the long days we had with you in Belgium when you were studying. So we have a very long and rich history together, Dr. No. And I'm also happy that you have taken this to another level, and I follow your activities online. Were it not for Corona, by this time I think I will be in India with you to celebrate this remarkable opportunity. I'm also happy to see my students have really joined into these meetings. The ones I'm seeing on my screen and the ones that I'm not seeing on my screen, feel welcome. I'm also happy to see attendees from India. And indeed, it's a big meeting. I can see we have 45 people attending this meeting this afternoon in Kenya and in the evening in India. So without taking much time, Dr. Nu, thank you so much for that introduction, and thank you for recognizing what we are doing. Of course, biofloc technology is now sweeping the aquaculture sector, and we have a lot more information that we still have not been able to learn. And every day new things are emerging. Every day we are learning new things about biofloc and about aquaculture. So with all that, kindly allow me to share my screen. Then I will move the presentation from there. Yes, you are right. In Kenya, we have a lockdown starting from 7. That's Kisumu City, where I am. The lockdown starts from 7. So I have to move to another town after this meeting. So I will request to finish early so that I can move with other people as well. Thank you so much. So I will share my screen.
Please.
Okay.
In the meantime, you start discussing. Yeah, please start the discussions, Eric, please.
Okay, guys. Sorry for that technical hitch. I'm not really sure why it cannot share. But Dr. Nu is sharing it in a while. So, ladies and gentlemen, Welcome to this presentation. Welcome. I'm joining live from Kenya. And indeed, we are talking biofloc technology. Kenya is still not very mature in regards to aquaculture technology using the new inventions. But I can tell you there are a lot of conversations currently going on. Interests are building, and people are trying to think now what technologies can we use to improve the production systems that are involved in aquaculture sector. And for us who are involved in the aquaculture sector, we have a lot of engagement with the ministry, with the government, and of course with stakeholders, talking the same information. How is it possible that we can be able to increase aquaculture production? Because that is the main issue. You remember, Kenya is a growing population. The growth rate is really high. We have a lot of challenges in terms of food and nutrition security. Almost half of our country is already affected by climate change. That is in terms of dry areas. And therefore, there's a lot of pressure for the small segment of the country to be able to produce enough food for the entire country. The population is growing fast, and people are increasing in number. Every day a mouth is born, and feed has to be provided. And we are talking about quality feed. Depanu, please, can you put presentation mode? So in terms of nutrition, then fish comes into the debate, and every food and nutrition security debate, fish has to be discussed. And that's why we are squarely into it, working together with other universities, working together with the stakeholders, working together with the government and research institutions, trying to understand how can we be able to promote. And of course, let me say by the onset that larviculture is one of the biggest impediment for aquaculture growth in Kenya. That means that there are no sufficient seeds for every farmer in Kenya. I think we are talking about a deficit of over 100 million fingerlings every year, because the fish aquaculture sector has grown. But again, now every pond, every farmer wants seed, and we are talking about quality seed for that matter. Where do we get the seed? The conventional ways that we use are already exhausted and cannot be used to produce sufficient quality seeds. So allow me to go into my slides. Deptano, if you are hearing me, can you put presentation mode so that I can start off? Yes.
Yeah. Is it visible?
I can see it, but it's a bit smaller. I don't know from your end, how is it?
Yeah, from my end is full screen.
Okay, that's good. So please move to the next slide.
Yes.
Move to the next slide. Okay, so in summary, this is what I'm going to talk about. I'm going to discuss about the fish larval seed nexus, and also the live food materials, how they are important in the, how the BFT is important in live food materials. Fish waste diet, this is a technology that we developed somewhere in Japan, and it's very useful now in Kenya. I will also talk about the live food dispenser, how we are able to even downgrade the scale to even use it in local ponds. Fish BFT in nursery ponds. The periphyton technology, that's the PFT, is also an emerging concept of BFT. I'll tell you in a bit how the concept and how it's being used right now. Then we have also noted through our research that the BFT actually improves seed quality and also suppresses early breeding behavior in tilapia. So these are new information that are coming in from research, and we are willing to disseminate them to our farmers and even practice them together with them. Next slide. So in terms of the fish larval seed feed nexus, ladies and gentlemen, I think this is where the problem is, because you look at the red rectangle where I've placed there. That is probably where the biggest problem is now growing to become a mature fingerling. That's where we have a lot of problem. And it's about diet, it's about management, it's about water quality, it's about handling. So, but for this presentation, I will dig deep into the feed aspect because that is also very, very important. So in Kenya, actually, we still experience larval mortalities to the tune of around 30% for tilapia. But for catfish, it is really, really high. Some farmers have reported up to 80% larval mortalities. Now, that is very, very worrying trend if that continues. Next slide. Yeah, so this slide is showing some of the live food materials that are very, very key for larviculture. And I talked about the feed or the food for baby fish. And in a nutshell, I'm talking about rotifers, artemia, copepods, and cladocerans. And you can already see they have different sizes. The important thing here is that the smallest live food becomes the most important for larval fish because, of course, for reasons that we understand. The mouth of the larval fish is small. It cannot be able to eat a big diet or a big particle. So the smaller the food particle, the better. And of course, remember, these are live diets. So they have a lot of nutrition. We are talking about enzymes, lipids, proteins, and all those stuff, even minerals. Very, very interesting. So if we can be able to shift our attention from using the nut diet to live food diet, then it becomes very interesting because then we are able to reduce larval mortality because we have sufficient live food materials for feeding our baby fish. And this is the direction that we are actually actively taking right now to be able to produce a lot of these live feed materials because they are actually being used to feed baby fish. Next slide. So one of the smallest rotifer was identified in Kenya, and this study is quite interesting because this was actually my study. This rotifer is called Brachionas angularis, which is slightly less than 100 micrometers. As precise as around 76 micrometers. When I compared it with the other rotifers in the whole world, it became the smallest. But then this animal consumes about 2.4 times 10 power 9 bacterial cells. That's where Biofloc is coming in. Remember, Biofloc is basically about bacteria. And of course, the optimal culture density that the larval fish requires about 10 rotifers per milliliter of water. This rotifer is highly nutritious, it is ingestible, it is digestible, and is very, very palatable for fish. So, of course, it's highly reproductive in a BFT system, and that's what we are going to discuss next. So if we can be able to, invent even more systems that can give us more live diet, then next slide. Yeah, so this is I'm trying to compare. On your left side, this is algae, thick algal pastes, very expensive, very, very expensive to produce. And on the other side, we have less effective inert diet. Remember, this is what most people use to feed their larval fish in the hatcheries. But what happens? Yes, the fish will eat them because they are hungry, but they will not be able to digest it properly. So at the end of the day, most of them will die. Now, that's why we require the live diet. Next slide. So this is a framework of what I did just to bring into perspective how we can use environmental wastes to generate bacteria in a biofloc setup so that it can be able to produce enough live feed materials. So I just use fish waste. Remember, fish waste are so many and so abundant. They say that about 30% of fish harvested goes to waste. Now, that's what some of those materials can always be used in a biofloc setup, mixing with molasses, with aeration, and some natural bacteria, maybe from yogurt or spoiled milk, and of course, with inoculation of live diet. And we can also add it with some chicken manure extract. I will come back to chicken manure extract in a few slides because of the interesting hormones that are there. Those hormones are very, very interesting. So the molasses there brings in the aspect of carbon source, and of course the natural bacteria from the yogurt and the spoiled milk, bringing the inoculant bacteria. So with the fish waste, remember fish waste is not being a substrate that's being used by the bacteria, and they grow in numerous, in billions of cells. So what happens? About 5% of the fish waste diet, that's through this protocol, you will get some organic matter, 15% of fine particulate matter, but the majority is microbial flora that makes over 80%. If you get this microbial flora, you can actually synthesize pure bacteria out of them. Now, this bacteria becomes direct food for these live organisms, which are being used by the fish as food. So that is the concept, that is the protocol, and this is the idea. And in the next slide, as Tanu shows the next slide, please show the next slide. We have been able to, use the same concept to, actualize it in the farms, and many farmers now own this. This is the next slide you are seeing here is showing how to extract chicken manure extract. Basically, it's a very, very, very cheap way to produce the liquid. Remember, you just take manure and you mix them with some coral powder for minerals. You boil it and you squeeze. Then you get soup. This soup can be thick or it can be not very thick. But you can also add add a little bit of pig manure, pig's urine. So this concoction is very rich in hormones. For example, the 17-alpha estradiol. We also have 17-beta estradiol and also estrogen. These are specific hormones that generate a lot of live food materials in a biofloc setup. So this is the secret we have been able to find. And we are able to use these materials to produce billions of cells of live food materials. And that improves the larviculture and aquaculture in general. Next slide. So this was another schematic flow. I call it a live food dispenser, to make it easier for farmers to understand. And this is just, showing how you are putting in the materials inside the tank, and this tank you can aerate, or you can also control temperature when you're using in the hatchery. And of course, with this, you can even put a tap. So you just remove a given quantity of water that contains a given quantity of rotifers, for example, or cladocerans, or copepods. And this technology, you are able to produce up to 3,000 individuals of rotifers per milliliter of water within 7 days. And of course, 1,500 individuals per milliliter of mixed live food materials. That is if you have rotifers, copepods, and cladocerans together. Of course, also within 7 days. And then after every 2 weeks, this system collapses and you have to redo afresh or start afresh. But then now what our farmers are doing, they are stagnating the production so that they have that continuous flow of live food materials from their live food dispensers. Next slide. Yeah, so basically this is the live food dispenser we are talking about in a hatchery setup. So some of our farmers already have this in their backyard hatcheries. So within this system, they are able to, inoculate the live food materials. They are able to put this fish waste diet, pig's urine, and the chicken manure extract, and this generates billions of cells of bacteria that are also food for the live food materials. And on the other side, like in this side, you're seeing the blue canvas tank. That's where there are actually the larvae, catfish larvae. So the farmers can directly transfer this water into the larvae fish larval tank, and this improves survival. It improves the fish veracity, and also it promotes the growth of the fish, and that's very, very important for aquaculture. Next slide. So, of course, we have also been able to document this information in terms of publications. You can move to the next slide, because these are actually just normal work that we are doing as the university. But then now, more interestingly, we can see here in this picture, the women are actually doing it themselves. This is installation of biofloc units in the ponds. Remember, most of our farmers still cannot get electricity connections, and therefore most ponds are just bare. So we are able to downgrade the concept of biofloc to very simple, unique things that farmers can really integrate with. And you can see in this picture, this was in Busia County, where we have an aqua park, park that contains over 100 fish ponds. It's bringing so many farmers together. And within here, we can put those materials. We have dead fish, can be put there, molasses. The woman is adding molasses there, cassava stems. We have also abattoir waste. Remember, when you go to the butcher, there are so many waste that are actually being thrown away. Those ones, you can bring them into the unit. Also with chicken waste, and even chicken manure extract. Now, this kind of unit is yielding billions of cells of bacteria that are actually direct food materials, or not only for even live food particles, but also for fish. What we have seen is that the fish are now moving towards the biofloc unit, and that also acts like a fish aggregation device. So farmers have found it easier, because then the fish can also be fed at the point where we have the biofloc unit, and the system is working. The farmers are recording very high improvement in terms of survival and also in terms of growth. Next slide. So yes, this is just a snapshot of the aqua park in Busia County, where we have up to 100 fish ponds, bringing over 200 fish farmers and fish cooperatives together. And we are currently installing these units in some of these ponds using the Kenya Climate Smart project that sponsored this project, the project that I'm running there. And of course, with other projects as well, we are able to bring these farmers together and even construct more ponds. So the interest is growing thick and fast. Remember, most of our farmers are still holding their fish fry in the ponds, and they grow in the ponds before they transfer them to other ponds. So we are doing everything inside the pond, and that's where we are also introducing this biofloc concept. The information is flowing down. Some farmers are picking fast, some are still lagging behind, but we are doing a lot of sensitization, a lot of training, so that they can be able to understand the concept, so that they continue feeding this system with the waste that even come from their kitchen to continue to maintain the productivity and also the production. Next slide. Well, of course, even in the laboratories where we train our students, we are also showing them how this system works. And also in the ponds, remember, you can see the green, the blue nets, harper nets. These ones contain some biofloc units, which are being used to feed directly into the fry or fingerlings. And this system makes it easier for the fingerlings to be harvested. So we have found a way of putting these units directly into the ponds where the fingerlings are, and also remove the parents of these fish, so that they can be able to consolidate and grow much faster and much quicker in weight. Yes, thank you. Go to the next slide. I think let me mention about the periphyton technology. This is an emerging frontier within the space of BFT. And we have just found out that it is possible to even translate biofloc technology in other languages or in other forms. So this technology is very interesting. One of my students is actually doing this right now, and we have seen very interesting results where we just put poles. They can be bamboo poles or normal timber into the ponds, and of course bringing in the biofloc concept, putting in the molasses and the feed and all that. The pole now becomes the central attraction point for feed materials. We are talking about biofloc materials, and you can see all fish now move to the pole, and they are able to scrap the pole to get all the nutrients that they require. The interesting thing is that this system, the farmers now use feeds that are low protein in nature, because most of the protein, and high quality protein for that matter, is already being produced within the ponds. So we find we are telling farmers that they can use even as low quality as 17% crude protein level of feed. And remember, feed is the most expensive part of aquaculture production. So if farmers can be able to reduce the protein content or reduce the feed intake, food provision, then that definitely translates into higher growth and higher income and higher productivity. Next slide. Yeah, a practical example is one farmer who is already doing it, and of course, with our supervision, you can see the pond there with sticks. And yes, one of my student is also doing this. And with this system, we are able to, see a situation where fish is feeding on 23% crude protein. That is cheap feeds. There's high survival, sometimes up to 100%. We have low FCR, and we are also experiencing faster growth rate, even as high as 5 grams per day. Of course, we have also seen that this technology limits prolific breeding. I will come back to that in a minute. And of course, there's what we call high survival activity index that I will also explain briefly. But quite interestingly is the fact that farmers are able to use less proteinous fish and equally get the same amount of product. And that makes it lucrative also in terms of economic viability. Next slide. Yeah, this is just to explain, the survival activity index. Actually, this just means how long can a fish fry be able to stay without food? I mean, during starvation. And this is an index that we use to quantify how stable or how strong a fish fingerling is. And we have tested that with Biofloc technology, the fish seed quality is enhanced, and to an extent that fish can stay even as long as 7 days without feeding. And that's very, very interesting. So you can imagine if we have strong seeds, then when we transfer them to the grow out ponds, there's highly likelihood that they are going to survive. And there's also high likelihood that we'll get good returns out of it. So this is just an index that we are using. As scientists, we are also curious to know what science is. Maybe I don't have to explain this, but what is interesting is the Biofloc technology can be able to improve the fish seed quality, and this can be attested using the survival activity index. Next slide. Yeah, of course, I think this is because we did this experiment already, and we have seen that with the Biofloc technology, the fish can survive up to 7 days without, during starvation. But look at the control. This is the normal way of doing things. This fish cannot survive for even 5 days during starvation. But then with in an optimal, this is now what the standard nature means, that maybe around 5. So therefore, it means that the Biofloc technology is also very important in improving the fish seed quality, which is very important for larviculture. Remember, this is evidence, scientific evidence, that Biofloc technology is able to promote the quality of fingerlings. Next slide. Again, yes, I promise that I will talk about this. In terms of prolific breeding behavior, this is something that we know that tilapia has this problem, that they start to breed while they are still very young, and that compromises the growth quality. Now, what the Biofloc technology has done is that, and this is periphyton technology to be very precise, we are able to see that the fish is breeding much later when you put it in a periphyton technology. If you look at the figures we have here, this is scientific information that the control will start breeding 4 weeks earlier. And then 2 weeks later, we are starting to see that the biofloc, the periphyton fish, starting to, to breed. So it is this delance that brings impotence, even in aquaculture. Why? Because then, remember, we are currently using 17 alpha testosterone hormone to sex reverse fish so that we can have all males. But then what this study is saying, with this technology, we don't need that 17 alpha hormone. We can actually perfect this technology, and then we automatically delay that breeding behavior. And of course, if we can be able to, this is still work in progress, what we are reporting right now, if we can be able to delay that breeding even by 8 weeks, then we can certainly say that we are going to achieve faster growth with the technology. And of course, we are also trying to explain some of these things are also still new to us. I think the fact that fish has to, to spend a lot of energy to navigate through the periphyton poles, so then they don't have enough space to breed. They don't have enough space to make nests. And that means that then they continue to grow. They don't spend their energy on the production. That probably is a possible explanation. But quite interesting indeed that periphyton is able to delay the breeding behavior pattern. This is really new to us, and we are looking at it going forward. Next slide, please. Yes, and even in terms of the primary productivity, and this is actually key, you can see we have a lot of rotifera and copepods in the periphyton ponds, and that means that that pond is very, very rich in terms of primary productivity. If you look at the control, which is the normal way of doing things, we are seeing more of the cyanobacteria. And look at this, very interesting. Cyanobacteria, this actually sometimes they even mutate to become harmful algal blooms, which actually affect fish life and survival. So what is interesting here is that in the periphyton technology, we are not seeing these harmful bacteria coming out as a sign that then the biofloc materials are actually resisting the growth of the proliferation of the harmful bacterial community, which is a biosecurity in nature. So it also confirms what we know in literature, that indeed the biofloc is also a form of biosecurity within the pond, because we are seeing suppression of harmful or bacteria that can lead to death or cause diseases to the fish. So this is quite interesting. We are seeing cladocerans is also high, and these are direct feed materials for the live feed. On the other side, we are not seeing much of that. Next slide. So again, finally, we did approximate analysis, just taking the flock materials and see what is the nutritional value of the flocks. And this is the result. You can see in the blue bands represent the periphyton technology and the control. And we are seeing a lot of proteins, up to 68% of protein. But then if you look at the control, we have more ash, which means indigestibility. And that explains, the significant difference, even in terms of fish growth that we see. So I think probably that should be the last slide, but if I can explain more. We are trying to encourage farmers to use local materials so that they can be able to improve the productivity in their ponds and in their tanks. And that we have seen happening. Our model farms are in Busia County, and that is where we are actually putting a lot of emphasis in terms of training, capacity building, and we are happy that even the government is now coming in. We have projects that are already funded, some we are running, some are already in the pipeline, and our next frontier is then to move towards intensification of biofloc technology, which I want to partner with my brother Deptano. He's already sending me some invoice and we are soon going to see how we can be able to intensify a biofloc technology so that we move not only just from fingerling production to grow out fish, table fish, and even explore other fish species, which are also very, very interesting for aquaculture. So, ladies and gentlemen, that marks the end of my presentation. Allow me to stop there and hand over this microphone to my very good friend, Deptanu. Deptanu, if you're hearing me, please take it over from me so that I can respond to any questions that may arise. Thank you very much.
Yeah. Thank you so much, Eric. I should say your presentation was not only resourceful, it was informationful. So earlier, many people used to think, in India especially, I will tell you, people used to think biofloc means making some tanks, putting some bacterial inoculum inside it and making big fish. But you have talked today that biofloc is a technology, the way you want to take it. Even you have shown down that how biofloc also can be encouraged in Southeast Asian countries where farmers are middle class income with low cost investment policy. So I am very much thankful, and I like to say my heartiest congratulations to you, because the way you are generating your energy into a comprehensive model of biofloc and taking into a nursery production and live food production is going to be a miracle in coming days. And in this context, I also like to take a small connections and like to tell you, our audience, you might be think that biofloc is a way of making bigger size of fish, but biofloc is the way also doing small nursery, hatchery management, live food productions, and also it is a way to make our production cost down, by which we can make our aquaculture venture more profitable, more economically viable. So I just like to take 2 or 3 questions from the audience side, because I know Dr. Eric got some other assignment. So we need some quick questions from the audience side. Be specific to your questions so that my friend Dr. Eric can address us, and any further information we need, we can connect Dr. Eric later on, because Aqua Doctor Solution from India and Dr. Eric University from Kenya, we are also coming up with some technical understanding of knowledge sharing platform, and this is the beginning of our first lectures. We will be coming up with such kind of lectures where farmer like me and researcher like Dr. Eric will be able to address many of your questions. Please ask the questions. Please unmute your self and just ask the question to Dr. Eric, please. If anybody want to ask any question, please go ahead.
Thank you so much for your presentation, Mr. Dittan Barman, and also Engineer Dr. Erickson. I am Engineer Achinta Kumar Mandal, calling from Odisha. So first of all, I would like to thank you, give you thanks to all the members. And I just need one small question from my side, that I am a new farmer, also maybe you know very well. We have just started a biofloc with 45,000 stocking of Elvanami seeds. So we just need your help, and also from your side suggestions that what is the real density required to grow in the biofloc tank for Elvanami?
Thank you, Mandal. Did you say Vaname? Is that shrimp?
Yes, shrimp. Yes.
Okay. For us, we still don't have. We have not started shrimp culture with this technology, so I might not be able to get the exact information. But that's the direction we are also taking. I know there's partnership from Jomo Kenyatta University of Agriculture and Technology, They are trying to, include shrimp culture for Biofloc. And soon we are going to Lake Naivasha, where we are going to collect the wild seeds to stock
Okay.
in some of those units. But for now, I'm not very clear about shrimp production using Biofloc technology, Mandal. Maybe
So
Deputy Amu can know.
okay, sorry for interrupting, Mr. Eric. So, do you think that because we know that in open pond we can culture the shrimp, but do you think that if we will culture in the biofloc tank in the new advanced technology, so it will be good for all of us?
Yes, indeed, Mandal, because shrimp is filter feeder by nature. So it filters materials, detritus, it filters bacteria, it filters even worms. So it's true that that biofloc technology can improve shrimp production, larval shrimp production significantly. That I can assure you. But of course, in Kenya, we still don't have practical examples to talk about in that regard.
Okay, thank you very much for it. Thank you.
Thank you, sir. Thank you, sir.
Welcome.
If anybody want to ask any more question, please you can ask to Dr. Eric.
Maybe, Dr. Anu, I need to emphasize a point that you are saying. In really true that in Kenya, most of our farmers are still very, very small scale, and then therefore they are not able to financially afford the intensive biofloc systems that requires energy, that requires aeration, and also requires special diet. So that's why we are also very careful, and we are taking it really to the lowest level that they can understand. And with this kind of framework, the farmers are building knowledge from the basic, and some of them are able to pick, and with the other support, they can be able to even increase the production to even up to intensive level by themselves. So indeed, we are trying to just make this information sink to the lowest farmer in the bottom line. And that's very, very interesting for our economy and for our aquaculture sector.
Yeah, true. Maybe I also like to emphasize some of our Indian friends who are into here. Maybe for them, I'll just make it a Hindi translation for 2 lines. With Dr. Eric presentation so that you can understand what Dr. Eric tried to address us. So I will speak in 2 minutes, though my Hindi is very bad, but I will try to summarize it so that Dr. Eric thoughts we can take it inside. Because if a person from Kenya addressing us very far from Indian, and if we take his knowledge and can implement it, we can do lots of wonders also because he's a very good researcher even. तो डॉक्टर इरिक सर ने जो डिस्कस किए हैं आज पूरा दिन में, उसका 2 वैल्यू आप में इसमें सुन पाएंगे। एक है बायोफ्लॉक टेक्नोलॉजी के माध्यम से हम लोग हैचरी में छोटा बच्चा के प्रोडक्शन कर सकते हैं, और साथ में लो कॉस्ट कम दाना खिला के, कम दाना का कॉस्ट उसमें डाल के हम उससे अच्छा प्रोडक्शन निकाल सकते हैं। इसमें 2 चीज सर ने एम्फेसाइज किया है, एक है बायोफ्लॉक सिस्टम, साथ में आपके पेरीफाइटोन बेस्ड सिस्टम। पेरीफाइटोन का अर्थ हुआ सर आप उसमें बंबू दिखाए हैं, पोल लगाए हैं, बहुत सारे चीज है जिसके माध्यम में अटैच एल्गे ग्रो होता है हमारे तालाब में। और सर ने जितने सारे चीज आपको मेंशन किया है कैसे आप बायोफ्लॉक तालाब के लिए बना सकते हैं, इसमें बात की है चिकन मेन्योर, हाउस होल्ड वेस्ट जो घर से खाना बच जाते हैं उससे बना के सब कुछ को मिला के कैसे आप एक न्यूट्रिशन बना के तालाब में दे सकते हैं, जिससे आपके तालाब में आप कम भोजन खिला के मछली के उत्पादन कर सकते हैं। और साथ में और एक चीज सर ने काफी अच्छा से बात किया, तेलापिया हम लोग जो लोग पालते हैं, हम लोग को हमेशा एक प्रॉब्लम आती है, तेलापिया 50 ग्राम 100 ग्राम होने के बाद ही तेलापिया अंडा छंडा चालू करते हैं। सर ने यहाँ पर बोला है इसका जो प्रोलिफिक नेचर है उसको बायोफ्लॉक और पेरीफाइटोन बेस्ड सिस्टम के माध्यम से जोड़ने से हम उसको भी अच्छा ढंग से कंट्रोल कर सकते हैं। सो I think if anybody still want to ask any more question, we'll take one more questions from side. Otherwise, we'll go ahead for the ending of the session. If anybody want to ask any question, please go ahead and ask to Dr. Eric.
Mr. Bora, I think you are muted.
Myself, Sri Osim Kumar Bora, working in the National Fisheries Development Board, Government of India, sir. Sir, this larval rearing of this tilapia species, what he is talking about. So in your country, sir, what is the survivability percentage from the spawn to fry or fry to fingerling? I mean to say in different stages of the life cycle, sir, in your country, sir, in comparison to normal system, sir.
Thank you, Bora, for that question. Indeed, I did mention that currently the mortality rate for tilapia is around 10, 20, going sometimes to 30%. That is if you use the normal way of doing things. And for catfish, we are losing up to 80, 90%, sometimes 100% fish gets, they die because of, problems within the system. But we have seen very, very important information, very encouraging information that some farmers have reduced the larval mortality by 10%. So we have seen a farmer somewhere in Nyeri culturing catfish, and he is now reporting 20% survival in catfish, 30% survival in catfish. That is very significant growth. That has never happened, because they are using, live food materials that are produced from Biofloc technology. Also for tilapia, what we have seen is that those who are getting fingerlings that have been produced using the Biofloc technology, those fingerlings are very hardy. They are very strong. They don't die, and they grow really, really fast. Remember, I talked about the survival activity index. So the higher the survival activity index, it means that fish is high quality. It will not die very, very easily. And that's high chances of survival. So all this together, we have information that indeed the Biofloc technology has improved larviculture sector tremendously and significantly. Thank you, Ashim.
Thank you very much, sir.
Thank you, Dr. Eric, and also thank you, Ashim Kumar sir, to raise a very nice questions from the farmer side, because most of the farmers also like to know what kind of things can be implemented. If anybody want to ask any more question, please ask. We'll take the last question from the audience side, please. If nobody wants to ask the questions, that's okay. I like to thank from Aqua Doctor Solution and Aqua One Center, Kolkata, Dr. Eric for a very useful, resourceful presentation of today, Sunday. And I think you make everyone Sunday today with your new concept of larva rearing process through Biofloc. And it will show a new path to many way, because we are also now starting with Biofloc based hatchery management technology in India. So I think by your knowledge, I also have learned something, so that which we can also take it into commercial level, and we can take it into next level of innovation in aquaculture. And also I like to thank the audience who ever participate from, even I can see somebody from also Vietnam and other parts of country, and from India also they have participated, outside the country also people have participated. I like to also say thanks to everyone for your precious time, and enjoy the session with us together with Dr. Eric. And we organize every Sunday 5 to 6 p.m. in India time, lots of aspects on aquaculture from globally experts and the experts from India and Southeast Asia country. And coming Sunday, we'll also have one young fisheries professional with just third year, but he is changing today Indian fisheries with digital platform, making lots of innovation through digitally. So be touch with us, because this is your platform, this is our platform, and we can all together make lots of innovations in the sector of aquaculture. And once again, thanks to Dr. Eric for your presence, for your time, and make this Sunday talk is successful with your contribution. Thank you so much, and stay blessed. Enjoy your Sunday, Dr. Eric. Thank you.
Thank you.
Thank you. Thank you.